Okay. While we wait for others, like I did mention earlier, let's just take to the chat box and tell us our names and the country from which we are joining today. Would really love that and would really appreciate it. I'm trying to set up the live streaming so that we can have. While we wait for others. Okay, I see the beautiful comments in the chat box. I see, like I mentioned earlier, Musa from Gambia. It's always a privilege to have you join us, Musa. Thank you so much. I see Oko Augustine from Babcock University, MBBS, MPH. Welcome, Oko. I probably had not seen you or seen this name, I should say, in the other sessions, especially Babcock University. I'm not sure I've read this in the chat box before. Perhaps you introduced yourself way after we had stopped reading out all of those. But it's nice to have you join us. Thank you so much. I really hope that you get great value from today's session and from the entire of the series as well. I see Nora from South Africa, a final year pharmacy student. Thank you so much for joining us, Nora. I see Eugene from Rwanda. Welcome once again, Eugene. Welcome, 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 welcome. Okay, let's keep them coming in, guys. Let's keep them coming in. Just to confirm, when we were entering the call today, were we asked to enter any passcode? Can anyone say something, please? You can unmute and say something if you're not going to be able to type. Please, were we required to type in any passcode or something? Okay, someone says no, and another person says yes. Okay. Okay, we just tried that to ensure that people do not have to stay in the waiting room and wait to join. Okay, okay. Eugene said yes. Okay, I think it varies. No problems. We just try as much as possible to make it this way. I think it's preferable this way to ensure that we don't have that restriction for people staying in the waiting room before they are admitted. So I think it's better this way. We just make the passcode as simple as possible for those who would be required. I'll just drop the I'll drop the link to the YouTube streaming on the WhatsApp groups now, so people can join from there as well. They are not able to join from Zoom. Okay, Musa, password was not required. Okay, no problems, no problems. I think we'll just make it this way going forward for the rest of the sessions. Okay, so just while we wait for others to join us, and while we, for those persons who just joined us, introduce ourselves in the chat box, your name, the country from which you're joining us, and any other thing you want to tell us, please feel free. I would assume that by now we would all see one another as a big family, right? A big family in the healthcare space, and trying to move forward the this area of medicine, precision medicine. So if we do at any point in time, perhaps even after the meeting, have any questions or need to make any clarifications, you can always reach out, please. I just thought to say that um, because someone reached out recently and um, the experience that she shared is something that I would never have thought of. So please and please, if there's any reason for you to reach out, there's an opportunity you've seen and you want to apply, or you think that you just want to get clarifications about it, um, please reach out. Do well to reach out, and we would be able to do what's in our best to support you. There's something we cannot do. There's not anything. Certainly, there's a lot of things that we cannot do. We have very, very, very selfless persons that we can reach out to. Um, a good number of them, the speakers we've had on this webinar series, that we can reach out to help out. Okay, so if you do have any interest, in fact, we've had persons reach out to say, oh, I would like to be connected to this person, be introduced to this person. How do I go about it? And we do what we can do to ensure that we get them connected. So the reason I'm saying this is, like I mentioned, the experience I had with the person who reached out. We are trying to put resources together to build and to support the growth of this field, precision medicine. And if there's one thing that we would have to do very, very well, would be very, very, that would be very, very required of us, it would be collaboration, right? It would be the fact that we are able to collaborate, whether on projects, whether on ideas, whether on the things that we do, um, linking to one person across the globe. That could be something that we collaborate on. I believe that and a lot more are part of the things that we need to do as far as this field is concerned, being a field that is still in its infancy, especially in the parts of the world that we are. I don't know. I can't confidently say this now, but I perhaps would be able to say with some level of confidence that the majority of us on this call are from low and middle income countries, whether Indian, I'm not sure about Pakistan, any country in Africa, and some other countries in the Middle East, right? So we share similar, I don't want to call it problems now, we share similar concerns about the disparity that exists in the field of pharmacogenomics and precision medicine, and in the field of genomics as a whole. You probably by now would have seen some of the content we've made about the World Health Organization 2025 global reports on genomics clinical trials that has been done since 1990 to 2024. And what that report reveals speaks loud. It speaks loud to the disparities that exist in the field of genomics research, its implementation, its utilization, and it just reveals a whole lot of things. And if I were you, and those of us on this call who perhaps have not heard about the report, the World Health Organization report before, it's just, I think, a 14-page report, very short. And when you read it, you get an idea about what researchers has been doing in the field of genomics, at least for those projects or research projects that were registered with the WHO between 1990 to 2024. So you get an idea of what has been happening. And if you're a researcher, you intend to be a researcher, it gives you so much opportunity because it just tells you this is where the gap is, or these are where the gaps are. These are the gaps that are needed to be filled. So I strongly do recommend that we go take a look at that report. It shows the different disparities in terms of age groups that are being studied, in terms of the countries that are participating in this research, in terms of the kind of diseases that are being studied, right? Non-communicable diseases are just the diseases that has been studied so far. So the communicable diseases, which is sadly the kind of disease that affects us in this part of the world, are given less attention, right? So you get diseases like cancer, and so many other rare diseases are the ones that really take the bulk of the study. Okay, Hiba is asking, where is this report? What I would do is, right, I would, before we end this call, I would drop a link to the report so we can have a look at it. I'll drop a link to the report. It's on the WHO website, actually. You can just go to head to the WHO website and search for it. Just type WHO Global Genomic Reports 2025, and you'd see that. Um, so the report details a lot of disparities, and for me, those are opportunities, those are research gaps that you can start preparing grant proposals for, right? Those are research gaps that you can start preparing proposals for, and look for bodies that give grants, especially in the area of research, in the area of genomics or precision medicine, and there are lots of them. And look for those bodies and start writing your grants proposals. A lot of us may think, oh, I'm in this country, and this country that I am in do not really have the equipment to do genomic research yet. You may not have to do whole genome sequencing or use all of those next generation sequencing technologies. There are still research projects that you could do with the resources that you have. Yeah, very true, Hiba. Thank you so much. I'm trying to see if our speaker is already in the house. I'm not sure about that. Okay. Let me try and reach out to him. Just a second, please. Okay. So I just reached to a speaker. Still trying to see if he's on the call already.
Hi, Destiny. I hope you're doing well.
Yeah, I'm doing well, Hiba.
Yeah, just I wanted to raise your attention about something. Since our speaker is from Saudi, I think today is the first day of Ramadan, so they are fasting, and I think right now is the time to break their fast. So maybe he will be a little bit late.
Yeah. He just
Yeah.
sent joining. He just sent joining
Oh,
now
okay.
to me on
Great.
WhatsApp.
Okay.
Yeah. Thank you so much for that, Hiba.
Yeah.
I really appreciate it. I probably should not have fixed the meeting for now because I know that a good number of us here are Muslims. I don't know how it works. Is it the same time for every country.
No,
The
no, it's actually different. That's why it will be very hard to manage at optimum
I
time.
see that. Okay,
Yeah.
okay. Prof has joined. Prof is in the house.
Oh, great.
Welcome, Prof. It's great to have you join us.
Thank you very much. You're welcome. Thank you.
Yeah, thank you so much. I just learned now that it's probably the time that you're supposed to break fast over there. Today we're really sorry for the inconveniences.
Right, exactly. I was having my, fasting, and then I was just actually having the food and just finished here.
All right, all right. Thank you so much. Thank you so much. So I'll just move straight to introducing our speaker now, since we're just some minutes behind time now. So for those of us who just joined, just use the chat box. We want to see your name and the country from which you're joining us. Please just use the chat box. That's really important, please. We'll move straight to introducing our speaker for today's session now. And ladies and gentlemen, our speaker for today's session is someone who has been with us, is someone who certainly has been with us and has been with us through the sessions we've been having, and is someone that is really an amazing speaker and an amazing person. Certainly, I'm sure you'd be able to testify to that at the end of today. So it's the 5th day of our pharmacogenomics webinar series themed Precision Medicine and Practice from Genomic to Real World Impact. And today, the topic we're going to be dealing on is bioinformatics, AI, and genomic data interpretation, what clinicians and researchers need to know. And ladies and gentlemen, we have no other person than my very own mentor, Professor Mohammad Liaquat. He's a consultant and a professor of pharmacology and neuroscience from Saudi Arabia. And Professor Mohammad, of course, is a PhD holder, is a distinguished healthcare consultant, academician, researcher, neuroscientist, corporate pharmaceutical trainer, and medical writer with over 20 years of experience spanning academia, research, and industry. His international research experience across several European laboratories has equipped him with diversified and advanced competencies in pharmacology, physiology, and neurosciences. Professor Liaquat possesses extensive technical expertise in sophisticated experimental methodologies, including electrofull feed potentials, CRISPR, Cas9 gene editing, AAV production, kindling models, histochemistry, advanced microscopy, and toxicological analysis. His translational research and data interpretation reflects both scientific depth and practical applicability. Beyond laboratory research, Professor Liaquat has served in senior academic and administrative leadership roles, including departmental head, chairman, dean, and director. In these capacities, he has demonstrated the ability not only to manage complex institutions, but to lead them strategically, driving organizational growth, academic excellence, and operational effectiveness. His leadership style is grounded in strong communication skills, critical thinking, time management, and a results-oriented approach. An experienced medical writer and reviewer, he has contributed to high-impact medical journals as an editor and peer reviewer, strengthening his analytical precision and scientific rigor. He is proficient in proofreading, copy editing, scientific writing, and research project design and management, making him a valuable asset in both academic and corporate environments. In recent years, Professor has expanded his expertise to include emerging digital and technological domains such as artificial intelligence, data science, digital marketing, SEO strategy, affiliate marketing, PPC advertising, WordPress development, virtual assistant, project research, product research, and motion graphics. This multidisciplinary skill set enables him to integrate biomedical sciences with modern digital innovation. Through sustained excellence in research, leadership, and continuous professional development, Professor Mohammed remains committed to advancing healthcare innovation, scientific education, and institutional capacity building globally. Ladies and gentlemen, I'd like to see the emojis in the chat box as we make welcome our very distinguished speaker for today, Professor Mohammed Liaquat. I'm not sure if I'm pronouncing that name correctly. Thank you so much, Prof, for joining us. It's really a pleasure to have you with us. Thank you so much. The floor is yours now, Prof.
Thank you so much. I'm really, kind of privileged because you have, introduced myself with an extensive introduction. So I'm really, thankful for this nice introduction. So you have explained so many things about my past career in the last 25 years. So as you guys have seen from the introduction, like I have a very diversified profile. So without spending any much time, I would like to share my screen so that
Absolutely.
it can go. Yeah, it is disabled now. So if you can give me the admin right, so I can share the screen.
Yeah. Just try sharing. It's going to ask for permission here.
Okay, still it is disabled.
Oh, wow, wow, wow.
Let me check now. Okay, now.
Yeah, I just made you co-host now. Please, you can do that now. You can check again now.
Yeah, it's perfect. Great, super.
Yeah,
All right.
thank you so much. Sorry about the inconveniences.
So can you see the screen now?
Yes.
Perfect,
Yes.
perfect. All right. So shall I start now or okay? Perfect.
Yeah, absolutely. Let's get to it.
So starting again, I'm very much thankful to you and for the whole team that has brought this kind of webinars series. It's really fantastic initiative, and I'm really impressed by the talent that Africa has, and I always impression that Africa has always has a good talent, and they really need more potential people so that they can work effectively and they can show their real talent actually. So, starting the today's session. So, as you know, the session topic is about how the clinicians, the researcher from the medical and the health allied field, how can they interpret the very typical field, which is basically bioinformatics and the AI and then genomic data. As we know, these days, all these new tools are getting more popularity and involvement not only at the level of drug discovery but also at the level of diagnostic, at the level of therapeutics, the treatment refinement. So as soon as the new tools are evolving, so it is very important for the clinician and the researchers that they get familiarized with them, at least to an extent where they can actually use them in a right way, because everything has a positive and negative things, and these are some technical glitches in that. So if you are not aware of those things, you cannot actually come up with the right decision for the patient actually, because we know we have a lot of decision support system nowadays. We have a lot of devices based on the machine learning, the AI, especially in a radiology, in a diagnostic. So we should always being people who are from the medical side, whether you are a doctor or a pharmacist or a nurse or any other allied health professional, you should always try to learn at least the basic of these things. And all these fields are different things. For example, when we talk about the bioinformatics, it's a whole domain actually, and within bioinformatics you have the neuroinformatics. So if you only referring to the specific field which is focusing on the neuroscience, the neurology, and the neurodiagnostics or neuroradiology. So you have the whole domain of the neuroinformatics, and they have the specialized databases, they have specialized tools. If you're not aware of those technicalities, you will not able to use them in a right way actually. So this is very important. You should get familiar with these things. When we talk about the AI itself, AI is a whole domain, the whole wider subject actually. So just like AI and the bioinformatics, the genomic field is itself a whole domain actually. So the topic is so extensive, I cannot actually give the basics of everything. So since as I know, a lot of people are actually they are the beginners, or they are either a student in medical college or a pharmacy college or college of nursing. So that's why I just actually finished the preparation of this presentation early in the morning, because I know like this is not a specialized audience. So that's why I have kept all the slide to a very superficial and a basic level, because if I started telling about the technical terms, technical things, and the tools, how the tools are working, it would be very difficult for you guys, because if you don't have any background about the basics of the informatics and the basic of the artificial intelligence and the machine learning and the genomic, how the genomics tools are working. When we talk about the any mutations, how can we detect the mutation, how can play with the antisense and the missense, and or any kind of peptide. So these things would be difficult for you to catch. So that's why I kept the topic very generalized, so that you can get at least the concept of this subject altogether when we integrate them all together, the informatics and then AI and then genomics actually. So in today's lecture, what we're going to cover is basically we can play around a little bit about the bioinformatics background, how we can integrate the bioinformatics with. Yes.
Prof,
Yes.
apologies, please. Sorry to cut short. Could we have it in presentation mode? Just
Yes.
for the
Yes.
sake of those that would be joining on YouTube.
All right. So can you see
Yeah,
now?
yeah, I think it's
Okay.
yeah, fantastic.
So
Thank you.
so we're gonna. Okay.
Yes.
No problem. No
It's
problem.
perfect now.
All right. So we're gonna discuss like how the bioinformatics can be chip in with the AI, and how can we apply them as a clinical practitioner to get the useful decision from them, and how you can actually verify the different modalities, the different model by training them, applying the AI and the machine learning tool, the vectors, so which are already in the pipeline. How can you actually use them, utilize them, and then report your own trend data, and then develop a model based on those AI and the machine learning methods actually. Then we're going to little bit discuss about the variant interpretation, which is basically genomic based. For example, UVS and the pathogenic way that can be useful in the regular clinical daily practices actually. Then we're gonna discuss about the integration of the bioinformatics with that of the WGS, which is whole genome sequencing, and the very new approach, which is called the multi-omics approach. And nowadays, actually, a lot of companies are working by combining all these modalities, combining the machine learning, applying AI tools, and then using the sequence and then whole genome sequence to come up with the right gene to address with the defective genes, because you have millions of genes. So what you can how can you analyze them in a fast, in a faster way? So you're applying the multi-omics approaches, and then you can come up with the AI assisted decision support system, so that you can apply the software in your hospital setting or your in a clinical setting based on these variety of tools, so that you can come up with a very precise and a very effective and a very quick decision making for the patient actually. And then we're gonna a little bit discuss about how it can be actually impacted the current practice. If you are a practitioner or a clinician or even a researcher, how can you basically apply them as a practical way? Because as I said, these are three different domains actually, and each has extensive different parameters. How can you chip in those parameters unless you don't have the background? So we're going to a little bit touch upon as a researcher, as a clinician, so that we can at least get to know the initial stages and initial step for that. And how can we define them in a real world practitioners as a in terms of the patient care, in terms of any research aspect. If you are going to design any study and want to apply any AI tool or machine learning tool, how can you use these kind of things, and because of the new development into the guidelines, which is given by the FDA, which is given by other national authorities, how you can apply the AI, because it's a new field. That's why the regulation were not developed fully. Now we have the very extensive thorough document about using the AI in the machine learning in the medical field. So if you are working in a research field, developing any medical devices, developing any tools based on AI or machine learning, so you have to keep those guidelines and ethical guidelines, which is given by the FDA or your own national regulatory body. So this is something which is very important always when the new things come. So the legislation and the regulation need to be defined, how you can use, up till when you can use, and if there is some mishappening, how can you basically handle them. So these are the major chunks of today's session. Let's see how it goes. So first thing, so what is the basic concept of bioinformatics? So bioinformatics is something, it's not that new actually. So if I go back, it could be like 40 to 50 years old story actually. So what you do actually, bio is simple mean life. Informatics is a combination of the tools that you apply in the biological system and to interpret them in an easy way. For example, you have a millions of sequence of the DNA and RNA. You have the lots of variant of genes and genomics. You have the different allele setup in your human body. How can you basically study them in an interpretable way? Because there is a amino acid sequence, there is a nucleotide sequence, there is a nucleotide sequence, there is a phosphorus group, there is a amine group, there is a three-terminal group, and then five-terminal, and how can you basically study them in an easy way? So you need something. So this is the tool, this is a field which can actually give you lot of information in a simplified way, so that you can interpret the any variation in the peptide, any change in the genes, any mutations which are basically in the sequence. You can easily detect applying these kind of informatics tools. So this is a field which can give if you give them a raw a sequence, if you give them any supposed sequence, so it can analyze the sequence, it can come up with the specific, because we have the once the whole genome is available in the system, in the software, it can analyze the defective sequence with that of the available normal sequence, and then identify where the missing sequence are, where there is the addition of amino acid, deletion of any peptide, or basically there is a mutation, or there is basically mismatching. So where it is happening, so all these can be done through applying these tools, which is bioinformatics tools. So you can easily study any gene expression. You can study the epigenetics changes in a body, in a system. For example, especially in the cancer situation, you can easily perform the epigenetics using different epigenetic tools. You have the proteomic approaches in which you can see the specific protein, because all the gliomics, like if you want to see the change in the glucose moiety, so you can do the gliomics. If you want to study any for example, the metabolic of any drug metabolism, because the drug metabolizes to the liver, so it is metabolized by different enzymes actually, which is called isozyme. There is a cytochrome P450 system in the liver, and they have a combination of more than 230 isozyme in that. So different drugs are metabolized by them. If you want to see and if you want to study these properties, you need the metabonomics approach. There are metabonomics too. If you want to specifically see only the proteins, there is a protein structure. If you want to see if there is any change in the protein level, then you can do the proteomics approach. So there are for example, if you want to see that there is a cell division, and you want to see there is any abnormality or there is any defective in the cell division, so you can also apply the transcriptomic approaches. So all these are basically the informatics tool, whether it's gliomics, metabolomics, genomics, proteomics, or a transcriptomics. So you can apply these things, and you can study at the peptide level, at the protein level, at the cellular level. You can see the changes, you can see the defect, you can see any variation in the specific time point. You can see any specific amino acid sequence. You can see any protein changes actually. So there is a protein misfolding, there is a defective gene. All these can be studied applying these tools. So you can see it's a wide tool that can help you to understand various things. So what generally, if you talk about from the research and R&D perspective, so what you do basically, so So you have a specific control, which is a quality control. What you do basically, if you see any kind of misalignment, if you see any kind of defect, if you see any kind of changes which is not normal, so you can, at the quality check level, what you can do, you can check the alignment, whether they are aligned or not. If there is a non-alignment, then that means there is a variation. So what the variation is basically, so what you can do, you can pick the variation, and then you can do the interpretation. Because for example, if there is a peptide, which is, let's suppose, take the example of insulin. Insulin is a peptide which is a long peptide chain having like more than 70 molecules in that of different amino acids. So if you pick any defective amino acid, for example, let's suppose at position number 25, so when you detect the position 25 with a defective amino acid, what you can do, you can interpret it easily that there is basically a change in the amino acid at this point. So you can easily transfer the correct amino acid and that point particular which was defective. So this is basically the very simple the flow diagram that we use when we want to study something. Or if you have the drug molecule, if you want to play with the different properties of the drugs. For example, if you want to increase the drug solubility, if you want to increase the viability of the drug, if you want to increase or decrease the excretion rate of the drug, so what you can do, you can play with the molecule of the drug. You can replace different amino acids, you can replace with different functional group, you can replace you can change the bond angle, the bond length, so you can actually play with the pharmacological property of the drug. All these you can see in a software simulation model. So when you change the structure of a drug by applying, for example, you can replace the hydroxyl group with the amine group, or amine group with the carboxyl group, and you can see the property, pharmacokinetic property in a simulation with what it will affect on the human system. So because you have play around with the molecular structure, and you are not directly using in the human, rather than you can simulate that, and you can see how it will impact the different body compartments, because we divide the body into different compartments. So one compartment model, two compartment model, three compartment model, and then see the drug dynamics, drug properties, the pharmacokinetic profiling of the drug we can do by applying these kind of alignment and variation changes, and see how the drug will actually impact the human system, whether in a positive way or a negative way. Then we have a lot of resources available. If you go on internet, there is a lot of, even in the PubMed you can go and see that there is a separate, if you, because you normally use the PubMed for finding the literature, you can also find the genomic sequence, you can also find the specific gene sequence, you can also find the specific nucleotide. So if you go on a PubMed and see, click the advanced search, you can get all these different or services which is actually as a database which is available in the PubMed. There are other resources available on the internet which is full of informatics tool, bioinformatics tool, neuroinformatics tool. For example, if you want to specifically work with the different neuroinformatics tool, you have SUS and BAMS. They are the different resource which is developed in USA basically. So you have enough resource available for that. So you have the reference genome, you have the different databases, which is nationalized databases. You have the database from the WHO, CDC, and national level. So all these are available actually. CDC is one of the very largest one. Then we have another database which can be, for example, the Sweden has a very sophisticated and very accurate database for their whole population. So you can use them and apply the bioinformatics tool and get different information out of that. You can predict a different disease pathogenesis pattern, you can predict any genetic profiling of a specific disease in that particular population. All these are possible using different informatics tools. Now we have it, as I said, like nowadays it's a trend. Now we apply the multidisciplinary approach. So what we do, we call it multi-omics integration, in which we apply the genomics, in which we combine with them transcriptomics and then proteomics. So when we combine different modalities, so it's called the multi-omics integration. So it could be, you can replace the transcriptome with that of the gliome if you want to study the glucose changes. If you want to see the metabolic changes, you can replace the transcriptomics with that of the metabolomics. So all these, when we combine the different modalities all together, so nowadays all the advanced level companies and R&D companies are, or the big sophisticated lab, they are applying the multi-omics approaches in different disease pathogenesis. It is very important we know the disease pathogenesis from the different angles. So we sometimes we study a disease as a system level, sometimes we study the disease pathogenesis as a cellular level, sometimes we do at the molecular level. Now we have to do with the genetic level as well. When we talk about the genome, we should know also there is basically protein, which is a building block. So we know the difference in the protein level as well. So we have to also integrate the proteomics level. We also have how the cell is dividing, so we also apply the transcriptomics approaches. So when we combine them, all these together will actually become a very effective picture that can give you an accurate and a very thorough information about any diseases. So another thing, this is something which is as we are doing all these things in a different computerized paradigm, in a software environment, in a AI based tool environment. So that's why something which you always remember in mind, which we see in a screen, the output, it cannot be sometime replicable in a clinical context. This is something we are doing on a software environment. So it might be replicable, it might not be replicable. It might be effective as we are looking at our screen, it might not be as much effective clinically as we are thinking. So this is the thing that you always remember with these tools, that they sometimes give you a very good model in a computer screen, but when we apply them into the animal level, into the animal models, or into the in vitro models, or in silico models, so they are not giving the same performance or result as we have expected from these informatics. This is very important. So the take home message for this slide is basically that you should always remember this about the bioinformatics as a short line, that this is not us, this is basically laboratory automation and a statistical layer of genomics. It's not something which is 100% clinically effective or 100% clinically accurate. This you have to actually prove at the later stage of research by doing the animal and the clinical research in the later phases actually. But this sets an initial stage for applying a systematic approach to get any ideal candidate, or to study any disease properties, or to study the behavior of the drug in a less consuming environment in a short time actually, as compared to the other. So why this is important, why we are saying like why we have to use a multi-integrated approach, why the informatics has been coupled with that. So as we know, as soon as we are progressing with the advanced medical researches, so we are come to know a lot of new things actually, and the data has been so huge. So if you want to study something, even simply if you go into the library and see the book of anatomy, or let's suppose a book of physiology or the book of pathology, like back in 1950s, it is very thin, few hundred pages. And if you see the book of physiology nowadays, a textbook would be like in thousands pages actually. Why? Because the research has been expanded. So when we are able to decode the human genome, so we know there are so many millions of base pair available in that. There are millions of nucleoside and nucleotide there. There is a billions of amino acid sequence in the whole human genome. If you want to study, can you do it in your whole life? It's very difficult. So you need something, some tests, some specific kits, some specific tools that can process the data so quick, so you can review them in months or weeks or even days actually. So this is important. This is the importance of these tools, that you can actually perform these kind of different genomic testing, whether it's at the level of exome or at the level of genome, the whole genome, which is WGS, the whole genome sequence, or just a panel, like you want to study specific couple of 4 or 5 genes. So this is called panel actually. So now there are various kits available, especially when you talk about the oncology field. There are kits available, there are genomic tests available. If you run those tests, you can easily detect the different kind of cancer. For example, one of the ideal thing is thalassemia, other is basically leukemia. So you can perform the panel and you can detect basically thalassemia, especially the beta thalassemia. These are the beauty of these tools, that you can, instead of studying a lot of things in a particular patient, you can just perform these specific genomic test and get the result very quickly instead of spending months and years in particular patient actually. Second thing, as we know, there is a huge data available. So from all over the world, the sequences are constantly reporting, and all these are sequences are stored in different resources available free of cost online. So if you want to read them, it's not that easy actually, because a lot of sequences available from patient, from normal person, from different geographical region, from Asia, from Africa, from North America, from European population. So there is a huge variation in the different disease. If you see the disease wise, so there is a different genetic variations. There are so many variants available. So you cannot actually take one of them and make as a benchmark and start researching on that. No, because there are, yes, so there are billions of reads that you have to actually do. So what, how you can do that? Because if you do not read the sequence from the available database, you cannot come up with the right variation in the genetic. For example, you take any genetic disease, so you have to replicate the specific sequence and then matches with the different databases. So this you can do easily if you have these kind of bioinformatics processing tools, so they can process your specific new gene. If you want to report a new gene, how can you know your gene is new? You have a database, your gene sequence will go into the database and it will match each available gene in that particular data set, and then it can tell your gene is new or not new. So if it is new, that means basically you have discovered something new in a particular disease state, so that you can report that I have discovered a specific gene pattern in a particular patient, which is the gene sequence is this and that. So how can you do that unless you have the validation of you have a resource which can be used to test your own gene, the gene that you are going to report. So that gene will go into that database, and that database will match every billion of sequence available in that, and then give you the result. Okay, your gene reported gene is new gene, so you can report the new gene. That this is something which you have to always remember. So that's why studying this kind of combination tool is very much important these days. Second thing, as the AI is taking lead in every field, so how can you do these kind of things applying the AI thing? So that's why we have a lot of new pharmaceutical, a lot of pharmaceutical companies using the AI based tools in the different diagnostics and the therapeutic purposes. So how can these pipeline will be coupled with the AI? So you should know the AI limitation, you should know the AI integration, you should know how the AI is being trained, how you can deploy the AI models, how can you basically validate these models, so you can aware of the AI things, then you can apply and you can enable your pipeline with these things, so that you can come up out of, let's suppose, 10,000 molecules. The AI based thing can give you 4 or 5 molecule which could be potential candidate for the particular disease actually. So this is something, unless you are not aware how this AI integration can be applied into your database of new molecules of the drug. So this is that's why it's very important, because as a pharmaceutical company you cannot spend now 10-15 years just to identify one compound. So you can do it now very fast within 6 months by applying the library of the molecules and apply the AI tool that can predict, okay, this molecule could be effective antibiotic or anti-hypertensive or anti-cancer drug, and they can start working on these instead of searching for new molecules out of 10,000 molecules. So they can do it with the AI based approaches in a very fast mode. Likewise, as nowadays we have so many, we have the lab test, blood test, which is from the different paradigm. We have imaging, we have basically other kind of tests. For example, in the cancer patient, you have the biopsies. So as a clinician, as a doctor, if you have a, let's suppose on your table like 10 to 15 reports together from the blood, serology, from hematology, or from imaging, an MRI, CT scan, and then biopsies. So as a human, you might take a lot of time to read them and then to make the conclusion. What the AI based bioinformatics tools can do is basically they can combine all the lab result, the patient history, and the images, and then within 1 second, they will give you the final outcome actually. But it doesn't mean you replace the human and the expertise of the clinician. No, it's just to facilitate, just to actually process the information quickly, so that the clinician can handle a lot of patient actually, because everywhere we have shortage of the good doctors, we have shortage of the nurses. What you can do, you can apply these AI and the informatics tools to streamline the process, to expedite the process, not to replace the human. So this always keep in mind. So these things are actually cutting down the time, streamlining the process, the medical processing and the management of the patient become faster actually. So this is something which you always clarify in your mind actually. So understanding their limitation is another thing, because as I mentioned, like in order to give the patient life the more priority, you shouldn't 100% rely on these tools. So you have to take your own empirical decision based on your own knowledge, based on your own experience that you have actually gained during your medical college, during your practice. Apply that knowledge with that of the AI based or bioinformatics tool based decision, and then give the final comment on that. Something which is very important. Never 100% rely on these tools. Always apply your own medical knowledge actually. So when we talk about the genomic based medicine, like it is another thing which is basically the concept of the precision medicine. So when we talk about the genomic based medicine, is basically shifting a very effective you can say the turnaround into the medical field. But the problem is it's so expensive. The treatment that you cannot imagine previously, you can treat the patient with genomic based medicine, but the problem is it is very costly. Not everyone can. A single injection in specific rare diseases, so it costs you only the one injection more than you can say $200,000 actually, and this one injection. So that means not everyone can afford that. This is something, but this is a paradigm in the medical field which is shifting a new breakthrough for those diseases which is not curable previously, like the rare diseases of human system, which is not if treated by anything actually. So you can only give the patient the symptomatic relief, or if you want to enhance the quality of life, so you apply like certain rehab techniques, but you cannot do more than that. But nowadays you can treat them actually. Imagine a person with a chronic genetic defects, and now you can treat them using these genomic based medicine. Not all, but at least there is a hope, because some of the new intervention being done in some of the very rare diseases, especially in the cancer conditions. So this you can actually now look at for those who can afford that. So the take home message for this is basically you as a medical doctor, as a medical practitioner, doesn't need to learn like how to code, how to program, but at least you should know what is basically going in the background when you apply these tools, how they are basically working at the working mechanism of these tools at the background actually, so that you can detect any kind of malfunctioning, any kind of abnormal results when you see the screen actually. So now how can we integrate the AI in the bioinformatics? It's very important, and it's very clear as we have already discussed a little bit on that AI basically is a tool that can actually make your life easier by faster processing of the information, by taking out the output in a more accurate way, by more precision with more accuracy. So this is how we call it the AI model development, and then model validation, and then model refinement actually. So in the model refinement, AI model refinement, what we do basically, we increase the performance of the model. That means that we are increasing the accuracy of that particular model. So imagine if you have a AI based detective system for let's suppose the respiratory infections. So the rate of respiratory infection detection, if we compare it, for example, is this for an example actually. So is if you do like manually using different lab tests and all these things, for example, the accuracy is, because we have always a chance of the false positive and the false negative in the result interpretation. So it is possible that the junior person is failed to detect the infection, the active infection, and he said the patient is not basically suffering from the respiratory infection, rather than he's just a mild cough actually. So this is something the AI can give more and a better accurate result compared to the clinical professional sometime. So there are a lot of studies on, especially in the cancer oncology field, that the performance of the AI tools is now surpass the performance of the clinicians actually. Second thing is basically when you talk about the tuberculosis, there are a lot of tools which has been developed which are more accurate than the human actually. Even one of the tool which is developed by one of the company which is called the Cure AI. So officially it is endorsed by the WHO for the detection of the the tuberculosis. So you can imagine now, just put the patient x-ray, chest x-ray, and then lab test and the basic information, and with a single click it can give you whether the patient is suffering from the tuberculosis or not. Imagine. So this is endorsed by, it's a pure AI based tool that can detect uh, you know uh, the active tuberculosis state actually. So I mean this I can give a lot of example, but because of the timings we have to finish the things quickly. So now we have the deep learning model. This is one of another layer of the AI. So we have the deep learning models. There are 4 effective model, but as I said, like I will not going to touch that because this is something very technical. So if you apply the deep learning models, different, there is a RNN, LNN, like convolutional neural network, which is a basic one. Then LTSM, if you are working with something with the voice, voice data, so you can use the LTSM, LSTM, sorry. And then you have recurrent neural networks, you have the convolutional neural networks. So all these you model apply to recognize the complex pattern, because sometimes you use a straight one, sometimes you need to compare a lot of different complex patterns. So for that you need the more accurate and the more deep down AI tool which is available through other deep learning models. So these tool can actually pick and suggest like if there is any pathogenic, if there is any variation, if there is a potential candidate that need to be taken priority. All these things can easily be suggested by the AI. So we have a large sequence and we apply the transformers. This is another AI based tool. And if you want to do more rapid detection, more rapid prediction, so we apply different transformers here. So these are called basically large sequence model. For example, as I said, there are millions of protein amino acids in sequences, so nucleotide sequences. So these is a very large database. So if you want to study the large sequence, then you apply the transformers. And then again, there are 2 types actually, in which we have the continuous one, and then we run them in a parallel actually. This is something which is AI is using in usages there. So whether it's drug discovery, target identification, repurposing of the the pandemic predictions, the the new molecule prediction of effective drug, and then you can apply the different tools for the diagnosis actually, and then you can apply in a refinement of the therapeutics actually. All this can be done basically. So when we talk about what the current and the latest trend that are integrating the genomics with that of the AI, so we have the very common one, which is not almost is like 20 years back. So it's called WGS, with whole genomic sequence, in which we can get previously when we want to do this, it costs like $40,000. Now you can do it like $100,700 actually. So you can imagine now how the technology evolved, and it is very expensive and very expensive. And the times goes and time passes, it become available for everyone. So still it is not that much expensive compared to when we talk about like previously 20-25 years ago. Now we have the new approach, which is basically the spatial multi-omics or the single cell omics approaches, which is if you want to study anything at the tissue level, at the cellular level, so what you can do, you can apply basically these kind of single cell approaches, and then you can predict any heterogeneity or any change in the architecture of the tissue or a cell architecture. You can detect with them. Then we have the another next step level thing, which is basically multi-omics approaches, applying the multi-model actually. For example, if you want to diagnose the Alzheimer's disease is something which has very complex pathogenesis. Some previously we were thinking it's alpha, the amyloid beta protein. Sometimes we thinking about there is basically protein aggregation, and then we have another thing that is the neurogenetics that tells, okay, the genes in the nervous system get actually mutated actually in the Alzheimer's patient. So all these are the different other things. So what now we are doing is basically combining all the modalities, the clinical approach, the imaging, neuroimaging, and then applying this precision approach for applying these genomic sequences. And then this is something which all together called the multimodal AI approaches actually. It's in different diseases, whether in oncology or neurology, cardiology, we can apply these multimodal approaches. But the very hot ones are basically oncology and that neurology. That is basically one of the very important area for applying the multimodal approaches in these two domains actually. Always you have to look at different regulation, and then okay, skip that. And then next one is basically explainable AI. Basically it's another thing which is a new, not very new, but a new concept when you talk about the domain of the AI. So it is getting more popularity. Why? Because it can give more accurate and more interpretable output, which is in a very easy way, especially when we talk about the predictive modeling, predictive genomics, predictive approaches. So this gives the more easy in a easy way the output of these explainable AI in a more easy or a user friendly way. That's why it's getting more popularity, instead of going through the deep junk of AI philosophies, like how it working, the deep layers of the AI, how they are cross matching things, all the layers are there, the hidden layer, then comes output. So it, the explainable AI gave the output in a very interpretable and easy way. So that's why it's getting. So the bottom line is basically is we need to, whether it's a student level or a professional level or whether you are a clinician, you need basically to grip of the AI literacy, especially from those dimension which is basically in the healthcare domain of the AI. So so that you can use them more effectively. When we talk about the genomic data, and that why it is essential, because as I said, like whether it's oncology or any other systemic pathogenesis, like from the cardiology or from neurology or nephrology or gastroenterology, each of these systems now have a clear link of the genetics. So when we are not aware of the genetics of the particular disease, how can you treat the patient actually? Because if there is a genetic defect, so it cannot be treatable by the drug actually. So now the genomic data interpretation helps you to pick up the defective gene or defective genome or defective nucleotide, so that you can work, and then you can come up with the new treatment modalities based on these abnormal genomic sequences. How can you change them so you have a new different techniques, such as the CRISPR, such as basically gene editing, such as basically the gene therapy, applying various tools. So all these things are the approaches which is you can say are effective in the future to treat any kind of genetic disease actually. So unless if your grip or background knowledge is very limited towards the genetics or towards the genomic interpretation, you will not come up with the right treatment for the patient actually. So now we have a clear guideline from different authorities, such as AMP guidelines are there. How can you basically classify the different variants? There is a gene variances. How can you classify the gene variances? Because they divided them into two clear distinct one, if there is a defective variance or change in variance, so they could be either basically the pathogenic one or a benign one. So if it is a pathogenic one, then you need to treat, and because every country has their certain medical regulation. So in certain countries the medical health is supported by the government through the national insurance system. So whether the doctor considered this particular disease towards the variance which is classified under the pathogenic stream, then it is subjected to a insurance. But if it is coming under the benign spectrum, then it is not subjected to a insurance actually. So this is something which makes the things clearer. If you are a practitioner, you can ask your patient like, if we want to go with the therapy, since this is only benign and you do not need any active treatment for that. So if you want to treat this defective gene, then you need to bear all the cost of that therapy. This is something you should actually inform the patient if you are aware of the different rules and regulation. So classification might be changed maybe next year, or that's why you have to look at the classification, which is as this field is emerging and transforming very rapidly. So that's why it's always important to look at the fresh guidelines for the national guidelines, or the main regulator, what they are saying about this. So there is UVS basically, which means basically the variants of the uncertain significance. Again, this is something which are very common, since they are not uh, producing any symptoms, or not producing any symptom that can deteriorate the quality of life. So that's why we categorize them as a UVS. VUS basically means variants of uncertain significance. So they are basically common, and maybe we might have this, and since it is not producing any symptom in our bodies, that's why we are not paying any attention. But if you do the genomic sequence, then we can detect this, there is a defective variance into particular gene, but it is not that much harmful, because it is not producing any symptom. So what do you have to, when we perform this genomic based surveillance or genomic based sequencing, then we detect basically these kind of variances into the population actually. So sometime as a clinician, sometime as a very early career professional, we are not able to interpret them. Why? Because the patient is normal totally. So but you do the genomic profiling, then you come to know there is abnormal amino acid or abnormal sequence there. So you can detect them, or you can mark them, you can spot them through the genomic tool surveillance actually. So this is something you should know, that there is another aspect which is called the VUS. You can also detect these UVS basically through the genomic interpretation actually. Then we have basically the phenotypic, this you can also predict, and you can also interpret it applying the genomic tools basically. So you have basically the gene, the G phenotyping analysis, and you can perform that, and then you can come up with the phenotypic differences into the patient actually. This is helpful when you have a combination of the genotype and phenotype, then you come up with the accurate disease symptomatology and the treatment modality. So applying the genomic as well as the phenotypic analysis, you can come up with the complete picture of the patient disease profiling. So the bottom line of this is basically you need the interpretation which is evidence based, and you need treatment of any complex system, any complex situation, applying the genomic result, because the genomic result is telling something which is not detectable through the routine blood test or through the routine laboratory test actually. How this is important slide, how the AI support the interpretation actually when we talk about the any disease variances actually. This is always, as I said, like the AI is a beautiful tool that can actually go deep down and analyze the available information and cross match this information with that available stored database, and then come up with, okay, we have analyzed your sequence and from available 20 million sequences from our database, and then we can because what the AI working is basically in the hidden layers. So if you put any sequence, it can take the sequence and analyze the sequence with the stored sequences, which might be in millions actually, each of them one by one. So this is something, if the human can do, it might take maybe 10-15 years actually to check one sequence, but the AI tool can do it very fast in a minutes, or sometime in hours if you are going through the whole panel and the whole genome actually. So this will take from hours to days and the weeks actually sometime. So what you can do with applying these machine learning tools, you can detect various sequences which are basically with the variances. So you have, let imagine a patient with a very complex symptomatology have, I mean, 20-30 variances which you don't know. So when you perform the AI tool screening, and it can actually detectable from the whole available sequence of several thousand peer databases available there. So you can cross match everything and then come up with, okay, So these 30 or 35 are the variances which is not normal actually. So then it is easier for a clinician to focus on these 35 variances and check which of the variances is a cause of the pathogenesis, because 35 defective means they are not pathogenic cause of the pathogenesis. Maybe 2 or 3 of them are basically cause of pathogenesis, and out of these 2 or 3, one could be the major one. So you can focus the major one, you can apply the gene therapy, you can apply the CRISPR technology to correct the defective gene or defective pair of gene. So this is possible through the AI can process this huge information of a defective gene through the databases and come up with the list of those who are basically in the variances in the sequences which are basically likelihood to cause the disease actually. So deep, as I said, the deep learning is more effective, powerful tool. It can give you more information in a parallel way. So you can apply the different depending on which kind of system you are going to work, what kind of information you are going to process, the data set, the raw data, what you are aiming for. Then you can pick the specific learning tool. So as I said, like it can easily predict the impact, what could be the impact of on the protein, on the system, its stability, and you can correlate with them on the available laboratory results, whether they are matching or not with that the predictive impact actually. So in journal, when you develop any AI model, you train them actually, and the training of the model is depend on the data set. The more accurate data is, the more robust and the more diversified data is, it can be well trained. And when the training is done well, that means the output would be more accurate actually. This is the basic key for the any AI tool. So if the data is not good, you will not be able to train them perfectly. You will have the accuracy might be 50%, 40%, or maybe low. But if the very good data, refined data, filtered data, and accurate data on a larger population, then you might get accuracy of 85%, 88%, or maybe 92%, which is excellent one actually. So all these things basically can be possible when we apply the AI tools in practice. This is for example, this is I put this slide for the purpose of the practice. If you read this case, I mean, everyone, I would ask you to read them, because how it is just a practice case actually. There is a 7-year-old boy with the development delay, and he is also having the seizures. And then he went for the WGS, the whole genome sequencing, applying the exome approach actually. And then the report came is tell basically there is a heterozygous missense variant in X gene actually, and which previously linked to epilepsy syndrome, and that link basically was reported in the literature. It is a cause of a epilepsy syndrome. And the AI score labels a variant as a likely pathogen. When you apply the AI tool, it is giving the final comment as a likely pathogenic with a confidence interval of 0.91, and which is like more a good one. Based on the multi-omic literature mining, the population data set, for example, the genome was absent basically, and the computational tool predicted there is a damaging effect. No, I mean, because you guys are not aware of the different tools, that's why you might have the problem in understanding. Because when we talk about the absence of a genome ad, this is tool. When we talk about the damaging effect, this is the terminology of the computation modeling actually. So there was no assay done on the patient, and when the family history and the family testing done, there is a variance found when we perform the de novo studies. So again, these are the technical things that if you are not aware of these, then it's not easy to catch this. And the phenotype of the patient is closed, but the perfect mesh need to be described syndrome actually. So the question is, for example, if you classify as pathogen, which is AI told this is a pathogen or likely pathogen, or a UV, basically VUS. So on what basis it is, it was classified? And second thing is, if you have, if you analyze all the scenario as a practitioner or as a clinician, So what the AI is predicted, that is the likely pathogen, or there was the population database is screening said there is no genome at, and the computational modeling said like there is a damaging effect. So would you convince with this, or you will have your own analysis? So if you have any thought on that, you can tell. Otherwise, I will move forward just because of the time. Because if I going to explain this slide one by one, each point from the AI point of view, from the computational point of view, from the population database point of view, from the variance point of view, which is de novo, it all needs explanation and then interpretation that might require more than half an hour actually. So you can this is why, that is why very important that as a medical practitioner you should have the background on these things actually. Anyhow, when we talk about the routine workflow in the clinics applying these bioinformatics practically, so so what we have basically, we have basically a couple of things that we have to keep in mind. For example, first of all, we should have the proper accreditation standard in that particular clinic or a primary healthcare center or a hospital actually. So if you are applying any practical tools or the bioinformatics, so you should have accreditation, or you should have basically ask the authorities to inspect your clinic or your hospital, whether we can use or deploy these kind of tools or not in our patients. So this is something which has to be taken into consideration if you want to apply. It's not like you, it's your decision, you, it's your hospital, that's why you are opting this. No, you should ask the authorities, you should ask the regulator or accreditation body, like I'm going to apply the different bioinformatic based patient tools, should I go for that or not? They will come and see and inspect basically whether your hospital has enough resources, enough infrastructure, enough data privacy and the firewall competency or not. Because the patient confidentiality is another aspect. From the ethical point of view, from the data governance point of view, from the cyber security point of view, from the logistic point of view, IT support and security support of the IT system, all these come basically when we talk about to implement these kind of system in our hospital actually. So this is something it is always that's why it is taking not only one time investment of taking for example buying the server, buying the software, and then running the software, but you also have the another component for the training your staff. When you train your staff, that means your routine clinical practice will be disturbed because you are going to spend time in the training and getting training for these tools actually. Third thing is basically if you train them even, are you confident enough that your staff is fully understanding these concepts or not, or they are started only using just by understanding 50% of the logic of this, and then they have started using it. So this might actually be harmful for the patient. That's why it is important, because of the complexity, because of the sometimes you have technical glitches, you have to report them immediately to the company that the software from where you have taken this one. So all these things is very crucial when you practically apply these things in your work setting at the hospital or clinic actually. And another important parameter is called basically quality matrix. The administrator of the hospital, the administrator of the clinic should review at least once a month the performances of all the tools they have adopted in their hospital. So everything, every parameter should be analyzed. If there is any mishappening, if there is any error, any mistake, it should always be reported actually to the administrator. If you are a nurse or a doctor and made any mistake based on these tools, just report them immediately, because it will help them not only to improve the system in your work setting, but they will also report it to the software company from where you have acquired this software. So they will also rectify this error into their software actually. So this is all basically, it's very important. So always the good quality matrix develops when you have a regular follow up, when you have a constant reporting on that, when you have basically constant audit of that every quarter, audit of all the log that you have used For example, you have seen in your ward like 500 patient, there are 5 doctors. That means 250 patient being used under the use of this software. So all these things should be ordered basically whether they have met the expectation or not. And once you are performing any kind of genomic analysis, it should be recheck actually, which is called the re-analysis, so that we can make sure that we have done the things correctly. And not only for this purpose, there is another advantage when you do the re-analysis. Sometimes when we do the re-analysis of the sequences from the patient, so we maybe in the first run we have skipped something, the bad sequence or a defective sequence, but when we reanalyze we pick that. So this is something it can give you the new information of the diagnosis when you reanalyze them. This is the second advantage of the reanalysis. So another important is basically those hospitals who are maintaining their EHR, the electronic health record. If you all the hospitals who are adopting these things, they are actually coupling or merging their EHR with these AI-based softwares. Why? Because they have the huge. If you are working in a tertiary care hospital and since the hospital is working since last 15 or 20 years, that means, and if they are maintaining the EHR for the last 10 years in their hospital, that means they will have millions of patient data in them. It's a tertiary care hospital, millions of data is already with the, if you integrate the EHR, as I said, the more the population data you give to the AI tool, it will be more accurate actually. So working in a tertiary care hospital, integrating with the EHR of that hospital, it will immensely enhance basically the performances of the software actually. And the clinician should know how they can request the reanalysis. So this is the genomic expert who do the reanalysis actually of the genomic sequences. All right, so okay, so this we have discussed about how can we integrate the multi-omics approaches, how can we do, there is another terminology which is called network medicine. So what you can do, you can combine all the protein, all the genes, all the biochemical pathways that are involved in a particular disease. For example, there is a biochemical abnormality in, for example, let's take an example of any metabolic disease which can involve biochemical pathways such as TCA pathway or HMP shunt or glycogenolysis in the diabetic patient. For example, let's take the example of diabetes. If there is a abnormality in the glycogenolysis or gluconeogenesis pathway, which are the biochemical pathways, so you can merge from one perspective. You only study the pathway, so you don't know what is happening at the protein level. And the proteomics level, you don't know what is happening at the biochemical level. You're only looking at the proteins only. At the genomic level, you're only looking at the genes. You're not looking at the pathways actually. What you can do, you can network all these together to pick the right framework for the pathogenesis of a specific disease, the key element of the specific disease, so that you can target those key elements to develop a treatment for particular disease. This is called the network medicine actually. Single cell multiomics we already mentioned in the previous slide. So these are the approaches which is very common, as I said, in the diagnostics. So another terminology is called neurotheranostics actually. So this is another new terminology, might be in some other time I can discuss about the neurotheranostics actually. All right. So now ethics, the we are now coming to the end of the presentation. So always when we talk about anything in the medical field, ethics is one of the very top level things. So we apply the genomic data, we apply the AI. What are the regulation of that? So as I said, recently the FDA have also released their guideline. WHO have their also guideline. There you might, your hospital or a national level guideline will be there. So you always look at and follow these ethics. You always able to protect the data actually, which is come under the data governance. That means it is not accessible by anyone. Even you should have a very strong firewall, so that is called under the cyber security. It should not be reachable by the hackers, because if the hacker will take the data, they will sell it to the dark web, because this is important niche. All companies who are working on that, they need the patient data, and the patient data is not available because of these bindings. So they need the data. What the hackers do, they enter into the hospital's server, and then take the data of the patient and then sell them to dark web. This is a job of a hospital administrator to make sure, ensure the data governance. It should not be available to anyone. It should not be accessible to the other people other than the staff which is on the hospital actually. So all these things are there. You should know as a medical professional, because some of you will become a hospital administrator or the owner of the hospital. So you should know at least the basics of the cyber governance, the cyber security that need to be ensured in your hospital to and safeguard the patient data of your servers actually. This is another important element. So I think this is a last slide, or teaching learning again. Okay. So skill needed, as I mentioned, you should know at least the basic learning about the what the genomics and the AI integration is, so that you can know how can you apply them in the diagnostic, in the prognostic, or a therapeutics. You should able to, at least critically appraise on the AI based test, because sometimes you as a clinician disagree what the AI decision support system is telling you. So you should report them by publishing these things. So this is something is very important, that you can able to comprehend, critically appraise on the AI tools which you think is not performing well actually. And then, since you are working in a team, you should have a very clear communication. What is interpretation of that? Because if the junior is working with you, he is new, he's not aware of the system which is employed in your hospital. So you can teach them, you can communicate them how you can interpret these AI based things which is employed in that particular hospital. So where awareness is very no, restricted or less, so what you can do, you can applying the local resources for the awareness among the patient. For example, you can mention there is a genetic counseling available, so you need to integrate with them with a genetic counselor. You also need to have a contact with the ethical committee, so that if you want to employ any of these things, so it has to be passed through first with the ethical committee of the hospital actually. This is something that clinic as a clinician you should also have skills to convince them why you are going to integrate this tool in your ward or in your department actually. So you have to convince the ethical committee, you have to convince the counselors which are responsible for that. This is very important. All right. So for the as a researcher, what skills that you need actually? First of all, if you are a researcher, you should know the basic workflow, the flow chart, how it works. For example, as I said, AI means first you need the data, and then you need to filter the data. Then you need to develop a model. Then you train the model. Then you validate the model, and then you deploy the model. This is all basically the workflow is. So what model you are going to use, what kind of data it is, all the tools are there, which tool is appropriate for this, how you will design it, how will you reproduce it. All these things basically is basically familiarity. So if you are a researcher, a student, trainee, you should know the basic the machine learning tools. You know which kind of method you will apply and integrate in the which kind of do you need the machine vectors, small machine vectors, or as SMBs or LVs, what kind of vector you will need in different data settings. All these things is basically at least you should know, because tools are available, but you should know which tool you need actually in specific. And then you basically applying the fair rule, fair principle, which is basically F for fundable, A for accessible, and I for basically interpretable, and R for reusable. So always apply this principle if you are a researcher in your lab, in your project, in your any proposal actually. So for the effective data management. And then you should have the collaborative skills. Sometimes you are not good in statistics, sometimes you are not good in computer skills, so you always integrate or collaborate with the other people, other department actually. So if working with the collaboration, always the output is effective, less error free, less mistakes will be there. If you do everything by yourself, chances of getting errors are more, and you might turn into a disastrous situation in some time. So this is something you should as a researcher you should know. So if you recap all these points, so the AI and the bioinformatics is something which is nowadays is getting popularity, and you will find them in the different levels actually, whether it's a level of a testing or a reporting. Clinically involved person or a healthcare professional, they should at least get the concept and the what the pros and cons, what the limitations of these tools are there when they are going to adopt that. Then we should also know that these kind of AI based systems, they are they are not something we cannot say they are totally perfect, 100% perfect. No, there are chances of errors in them, there are mistakes in them, so we have to always keep this segment about the AI tools, that they might give a wrong result, they might give a wrong interpretation. So always doesn't take them for granted. Always actually, always give yourself enough scientific depth to reanalyze what the output of the AI based tool is, and then apply your own knowledge and then come up with a decision, not purely on the AI based decision. It's an emerging trend applying the genome, WGA, the multiomics, and explainable AI. So this is something which is transforming the diagnostic and research in the coming years. Always we have to look at the patient concern regarding their confidentiality, regarding their the privacy. This is our job to safeguard their information as per the guidelines. And the last point is basically we cannot do everything ourselves. We are not perfect actually. We are not superhuman actually. So we always combine with others. We develop a network with others. We collaborate with others so that we can come up with a better outcome. So this is all from my side. And if you guys have any questions, please ask.
Amazing, 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 amazing. Thank you so much, Prof. I saw it meant in the chat box. Thank you so much. It's been a very wonderful presentation, and I can see that we already have some hands raised up, and I see some questions in the chat box. We're just going to go straight to taking the questions now. Thank you so much, Prof. We'll take this one in the chat box first, since it came in first, from Adebukola. Adebukola. It says, okay, there are three questions here. How can I? Okay, let me just read them out, Prof. Perhaps you'll be able to just answer the three of them. The first one says, how can I test the accuracy of artificial intelligence? And the second says, are there ways artificial intelligence can be used to interpret variants? And lastly, it says, is there any ethical guideline for the use of AI in genomics?
All right, so very nice question. First of all, the first one basically, how can you test and validate? So I can give you one bigger example. So the largest AI tool that is very common, everyone is using basically Chat GPT, which is from the OpenAI company, one of the largest company, OpenAI. So there, the latest, because I'm constantly using and testing their performances since its launch. So the performance of this is called the AI hallucination. That is to generate the fake information actually. That means against the accuracy. So it was like 40 and 30 to 48% in the beginning, and now when they have improved it with the Chat GPT 5.5 and integrating DLL dot technologies, now they have reached to 60% actually. This is the largest one because I'm talking about the LLM, which is when we talk about the machine learning. So we have NLPs, we have LLM basically, the larger database level we are talking about. So imagine when these kind of big tools have the accuracy of 60%, because they are using the data from billions of data points. So that's why it's a very huge data. So performance of 60% is still like a good one. But in medical science, if you say 60%, that means 40% patients are basically wrongly diagnosed or wrongly treated. This is something. So that's why, coming up with the direct question, we have a specific disease based diagnostics tool which are having the efficacy, or sorry, efficiency and accuracy of the 88 to 92%. So if I tell you the performance of that the Cure AI tool, which is diagnosing the tuberculosis, is basically 95% actually. So this is something which is very, very promising. So there is no rule of a thumb. Whatever AI tool you develop, you have to basically set the accuracy by your own. So there is no journal thing, because it is something when we manufacture any car. So the engine of the car, each car is tested by the company. The same applies here. So if you develop any AI tool, you have to see the accuracy, you have to see and judge the performances. And if its accuracy is very low, then you keep train the model until you get the accuracy of more than 85-88% actually. So the companies who are launching their tools, they already made their AI based system and train them to a level which has having accuracy of more than 90 or 88% or 85% actually. So this is one. So the second question was, what was second question?
He says, um, are there ways that artificial intelligence can be used to interpret variants? That was the second question.
Okay, so actually there are various category of the AI tools when I talk about, because if you are only specifically want to check the variant calling in the genomics, there are tool available actually which are only focusing to check the accurately the sequence from the data, only check the variances basically. So for example, if you want to see the from the pathogenic or a benign aspect, so there are multiple features in that available. So what your main objective is, you have to use that particular tool and then mark the tool with the particular feature so that it can pick. As I said, it's a larger database. You want to see the protein, you want to see the function, you want to see the structure, you want to see the single cell, you want to see the transcriptome, you want to see any other thing, RNA sequence, DNA sequence. You want to see, for example, the nucleotide or nucleoside. All these things have. So if you will not pick anything, it will take a lot of time for the processing. So if you want to only check the variance, so there are only specific variance based tool that can only detect in a sequence the particular variance actually. So if you want to detect the sequence in the whole genome, it will take longer time. If you want to check the variation in the any new gene from the patient whom you are treating, it will take less time, because you only took a particular 7 or 8 or 10 or 12 sequences to test them whether they have the any variance or not. You will actually run the database and you will actually come up because you have only 10-15 sequence. But if you want to have from the whole genome, it will take a longer time. So all these things can be done through specific softwares tool that can be used. So there are available different softwares that can be effectively used in that. And then the last one, it was, is there I think if I remember correctly, you asked like if there is any specific tools to detect which is in the bioinformatics.
Ethical guidelines. Yeah, ethical
Ah,
guidelines for the use of AI in genomics.
yeah, actually, if you see the recent guideline from the WHO, is a first extensive draft which is released nearly 3 or 4 months ago by the FDA. So it is an extensive one in which there is a division actually. So if you are working on a device which is based on bioinformatics, there is ethical parameter there. If you are working with the biologicals, there is a guideline there. If you are working with the drugs, there is a guideline there. So each segment is covered there with the ethical guideline, with the patient safety guideline, with basically regulator guidelines. So you can go through that. You can, if you go on a FDA side, you can find that. So beside that, there is also a guideline from the WHO. So you can also find out. Let me check if I can share you the link here. If I wait, you give me a minute. So if I can find the link here. Hmm. You can ask in the meantime the next question so that I can type and search.
Okay, absolutely. That's very fine. I think we can take Amadu. I think Amadu was raising the hand first. Amadu, you can unmute and ask a question. Amadu Ayedeji.
Thank you very much, Pharmacogenic. Thank you very much, Prof. Thanks for the wonderful presentation. By name, I'm Ismail from Nigeria. My question is, you talked about simulation test software. I am asking that is the software available for everyone use? So, and my second question, how can someone access the library of molecules so as to, like, if you are doing the gene variant, how can you access the library of molecules? Those are my two questions. Thank you.
All right, all right. So where is, okay. So here I am pasting the link. So you can go and read this guideline of the FDA that you were asking about actually. So there is a segment there, you can get the segment in particular. So this is the main link, and then you can browse back, you will get a lot of things there. Okay, and then wait, about the WHO, where is that? Big data. Okay. So if you want to test anything in the children, they have the separate guideline. You can download this PDF here. All right. It is an ethical guideline. If you want to apply, then you have to pass through the ethical guideline from your institutional the review board actually. So yes, and then there is another one. Again, life cycle. No, this is not the relevant one. All right. Okay. So the other question was about like the tools they are using. Actually, I can also give another link. I can tell you format. So that if you want to look at any nucleotide, nucleoside sequence, so you can look at here free of cost. So if the link, so you can look at the link and then so all field, if I go from mesh, here it comes sequence, nucleoside sequence. And then there is okay, I can write it here. So there is another database which is called CC Wonder database. So this is a database which is freely available. You can, if you can Google it, you can found this one where you can get different resources, which is freely available at the country level, at the regional level. So you can get a huge data from there actually, population wise. So this is one of the resource, and wait, I can send you another link for all right. So this is all right. So this is another important where you can easily get the, not only you can visualize the genomics, but you can also get basically the regulatory element, and you can uh, customize it and then play with that. And then we have if you want to work with the protein protein interaction and see any difference in the protein functioning, So you have basically this if you read the paper on that, lot of people get the database from their string, which is called the string database actually. So they get it and then they start again. Then we have basically another which is called igb.org. So this is another database we can look at that. And the major one I was telling you is about basically PubMed. It's PubMed Blast. NCBI tool. If you go on a PubMed site, so they have the different, as I said, the gene, the nucleotide, the nucleotide specific protein. So you can go. NCBI is basically the PubMed, National Bioinformatics tools you can get. All right, so you can get it all these things from there actually. All right, so gene bank. If you click this link, so what you can do is you can basically get the different, for example, nucleoside only genome, the geo database, the geo profile, nucleotide, geon. I mean, there are more than 20 to 20 different subfields you can get it there. So just click this link and you will get your required things. All right. I hope this will be useful for you guys. So, and then GitHub is one of the largest one where you can get everything actually. So, and for the article writing, so this is the largest one and everybody use is basically GitHub. Okay, there's so many questions here. Yes.
Oh, thanks, Prof.
All right. So next question.
Okay. I think we can take that from Faith Olorunwa. She says, I want, Prof's view on having a dedicated AI, say
Any other
healthcare
questions?
AI, which, can you hear me, Prof? Hello, Prof, am I audible? Can you hear me? Oh, I think the network might be. Can you hear me? Am I audible? Oh, wow. Okay, you can hear me.
We can hear you clearly, yes.
I can.
Yeah.
Okay,
I
can you hear me now,
hear
Prof?
you.
Can you hear me now?
I don't know.
How about now, Prof? Am I audible now?
But I cannot hear.
Oh, wow.
Sara Al-Muthari. Yes, I have some questions. Yes. Can someone else use the mic so that I can know whether I can listen or not? Because I cannot listen to the speaker, the moderator mic.
Please, someone else
Prof,
should
can
say
you hear
something,
us?
please. Okay, great.
Prof, can you hear us?
Good evening, sir.
Okay.
Oh, wow. I think.
All right. So you can see the recording then.
Yeah.
Sara
Can you hear me
see
now,
the
Prof?
recording later on.
Can you hear me now, Prof? Oh.
It's not the course, it's a series of lectures. Let me re-log in actually. Maybe I can listen then.
Oh, okay, Prof. I think they can hear you. Can we hear Prof? If we can hear Prof, Prof can just take the questions from the chat box directly.
Yeah, we can hear Prof.
Okay, great. Prof,
Yes, sir, we can hear you.
if you can hear me, Prof, you can just take the questions from the chat box directly if you cannot hear me. Let me type in the chat box.
On a stream, I still okay. I am.
Okay, Prof, can you hear me now?
See the screenshot. Okay.
Oh, wow. Let me stop sharing my screen.
Okay.
Prof, can you hear me now?
So in the chat box,
okay, yes.
the previous message has been gone. Can you just take the question directly? Yes. Thank you, Sarah.
The network is really funny today.
Why am I? There is no more text in the message box. Actually, when I, how do we get the certificate attendance? Attendance register.
Can you hear me now, Prof? Oh, wow. Network has really been crazy today, like from the start of today. Prof, can you hear me? Amazing. So yeah. Prof, can you hear me? Wow.
Attendance link for today.
I don't know whether I could just tell Prof the question you're trying to bring in, so that it will not be wasting time.
Exactly. I'm not even sure if Prof can hear you. Please just try speaking up if Prof can hear you.
Prof, can you hear me, please? Good evening, sir.
Oh, I'm not sure he can hear you. Probably the network over there. So I don't know, Prof has shared a lot today. Like the resources. In fact, I didn't expect this myself, and we are very, very grateful and going to recommend and strongly encourage all of us to ensure that we look at these things The video is going to be uploaded to, in fact, it's been streamed to YouTube now, so you can always go back to look at it and then make sure that you make the most of these resources that has been shared. I don't know how it's going to go, but we're going to have to call it a day now. I just shared the link to the attendance now. Just before we call it a day now, let's just take some few announcements. And the next session we're going to be having is going to be
possible meet at least 60% of total attendance.
Okay. Prof, can you hear me now? Oh, I don't think Prof can hear me. So so the next session is going to be on the 24th, of this month, 24th of this month, and it's going to be by 7 p.m. GMT +1. So that's going to be 2 hours from the start of today's session. So that's when we're going to be having the next session. And on that day, we're going to be learning practical stuffs, right? We're going to be having practical stuffs. We're going to have a pharmacogenomics researcher come to talk to us on that day, Nicole Kula. Perhaps those of us from South Africa would know Nicole Kula. Perhaps even those of us who have been, who know about PGRN, Pharmacogenomics Global Research Network, you probably would have seen some of the posts from Nicole. She's a very loud voice in the pharmacogenomics space. And on that day, we're going to be navigating open access pharmacogenomics resources, and we're going to be having interactive case sessions. So, in fact, I just got an email from her this afternoon about she was thinking of including some other things that would also be beneficial to all of us. So as soon as I'm done with this session, I would have to respond to that email to ensure that we get the best on that day for all of us. So you do not want to miss that session, because all of the things that has been gone through from the start of this session till this moment is going to be unraveled in terms of the practical aspects of it. How do you actually use these resources like CPEC, like from GKB, and the ClinP, which is now ClinPGX, to actually inform clinical practice? You do not have to actually have all of the resources, all of the machines, the equipment to be able to actually do some pharmacogenomics. So with those resources, with those open access pharmacogenomics resources, you can actually be able to practice some pharmacogenomics in your facility. And then lastly, we're going to be sending a survey to the WhatsApp platform, as well as the to the registered email, the emails we registered with. And the survey is basically to get an idea of how these sessions have been helping us. And we're going to need your various to the surveys we'll send. Please and please. Ability to be able to get certificates at the end of the entire series is 65%. Please, let's ensure that we make these sessions. And apart from making the sessions, let's ensure that we are making the most of the experience. Okay. Prof, can you hear me?
Yeah, I can hear you now.
Oh, great. I don't know if you still have some time to spare with us, because I probably would. I know some persons still have hung around now to be able to get response to those questions. I don't know if you have some time to spend with us. Can we take some more questions?
Yeah, sure. You can take, no problem.
Oh, great. Thank you so much. Faith asked a question earlier. Faith Olorunwa says she wants your view on having a dedicated AI, say healthcare AI, which would only be for health-related data. How far will this help in improving accuracy?
Even head related? Okay. Is it head related or what?
Yes, healthcare related.
Okay. Healthcare overall. Okay. I mean, there are so many devices. I can tell, like only last year, so FDA has approved more than 364 medical devices actually itself. So the field is emerging. So there is not one, but there are several. If altogether we look at the data of the FDA, only FDA, we are not talking about other countries, because for the European regulatory authority there is a EMA. The Australian regulatory authority is different one. So only if you talk about basically FDA, they have like only last year they have approved in term of devices which is AI based is more than 370 actually. So the field is emerging, the field is growing. Even there is a recent development in term of which I've shared in the one of my group, which is also the your moderator is also part of it. So there is a AI based device for the treatment of the depression actually. So in which there is a specific stimulation goes to a brain. If you use that device, so you will actually relieve from the depressive episode actually. So this is something the field, as I said, is growing. It's not one. There are so many, several one. There's a device for the migraine. So there is a lot of, but if you take into consideration the proportion of the system wise, so out of those 374 devices, 60% are from radiology side. So radiology is something which is more the hot domain for the AI based discoveries, AI based devices, and AI based diagnostics actually. So in the coming year we can expect like the number will grown into a thousands actually, in which you can have a AI based lot of softwares. There is a company who has recently launched app which can detect basically more than 14 major skin diseases. So this is something which is purely AI based. With the help of your mobile camera, if you download the app, so it can detect the skin different, like for example eczema, urticaria, and then pruritus. All these condition can be detectable by this application, which is basically trained on several thousand of patient data, and it can detect this with more than 80% accuracy. So many devices in place actually. And then as I said, like we can expect by next year the number will be grown up into thousands actually. So yeah.
Okay, great. Thank you so much, Prof. This person's asked about your contact. Amuda, I think Prof is very active on LinkedIn. You probably
Yes,
would want to connect with Prof on LinkedIn. Yeah, Prof is very active on LinkedIn.
yes,
Amuda,
exactly. You can contact me
all
on
right, great.
yeah, exactly. I'm not on
Great.
other networks because I don't have much time to. I'm not on Facebook, I'm not on Twitter, I'm not on Instagram, nothing else. So yeah.
Yeah, great. Thank you so much, Prof. I think that's all of the questions that we have here in the chat box. And yeah, someone just mentioned the day three.
Mhm.
Someone just mentioned the day three session. Thank you so much for the reminder, please. In fact, I just forgot about that now. Yes, we're going to be having the session. I got an email from Professor Kazim yesterday, just yesterday, reverting that he's going to get back to me on a suitable date. So we have to reschedule that date because of course, what happened the other time. So we have to reschedule that date. So it's probably going to be after 24th or before that. I really don't know for now. But as soon as I hear from Prof on a suitable date, I would certainly reach out to us on that. Thank you so much for that reminder, Gina. Thank you so much. Okay, I think that's all for now. Thank you so much, Prof. Thank you so much. We are
My
very
pleasure.
grateful.
You are welcome. You are welcome. Thank you.
It's been such an impactful session today, like so impactful. We are very grateful, and we continue to hope that the efforts that you've put into all of this will continue to translate into greater good for our world and the entirety of humankind. Thank you so much. Thank you so much.
My pleasure. You are welcome.
All right. And that is it for today. Amazing people. We're going to be doing this again on the 24th. Do not forget, on the 24th. And it's going to be 7 p.m. on that day, 7 p.m. GMT +1. Please, let's ensure that we mark our calendars for that amazing session. Certainly do not want to miss that. And I would advise that you join with your system. For those of us who have systems, I mean desktop system now, not mobile phone. If you have your desktop system, I would advise you join with it on that day, so you are able to actually practicalize what would be done on that day. You can go to the website, the resources that will be shared. You'll be able to use all of them and see how they work on your own end. Okay, that is it for today. It's great to have us all join. See us all on the 24th. Thank you so much, Prof. Thank you so much. We're calling it a day now. We are very grateful.
Thank you so much, you and everybody who are listening to me. Thank you very much.
All right. Thank you so much.
Please, before you go, your handle, please.
Okay. I would drop that. Let me. You know what? You can just, the name on the flyer. Just type the name on the flyer on your LinkedIn, right? Just type it on your LinkedIn search bar. Prof's name would appear. You'd see his name there. And I think it's the profile picture that's on that profile that we have on the flyer, so you can easily recognize it from there.
Thank you very much.
You're welcome. You're welcome. All right. Good night, everyone. So as I mentioned earlier, if you do have any questions or anything at all that you think you would want to reach out to us about, please, our DMs are always open. We may not respond immediately, but we would certainly respond because, of course, I'm a student, and a lot of us here at Pharmagenosphere are students as well. So, and we're pharmacy students, so it's a lot of things to combine that with doing all of the things that we do here. So we do not respond a day, 2, 3, 4 days, we certainly would respond. Be patient with us. Thank you so much. All right. Good night, everyone. Bye-bye. Good night, Prof.
Good night, everyone. Good night, Prof.
You too. Good night, everyone.
Good night. Bye.
Bye-bye.
Bye-bye.